Hello dear students, welcome to Biochemistry by Revati. I am Revati and in today's class we will learn an important topic that forms the foundation for many biochemical processes and the topic is weak acids and bases. I guess you will see these concepts again and again in your study of enzymes, buffers and metabolism. So pay attention. Buffers we have already completed in the last video. If you wanna check, I'll give a link in the description. Please go and check out. Today's topic is weak acids and bases. Let's understand them deeply today. Let us study now what are weak acids and bases. Acids are the substances that donates protons, and the bases are the ones that accept protons. But not all acids and bases behave the same way in water because some dissociate completely while others dissociate partially. These were the terms strong and weak come into play. So let's see weak acids. Weak acids are the substances that do not ionize completely in water. Example, acetic acid. Acetic acid dissociates into acetate ions and hydrogen ions. So here acetic acid is a weak acid. Acetate ions are acting as a conjugate base. Next, weak bases. Weak bases are the ones that do not produce hydroxyl ions completely or do not accept protons completely. Example, ammonia. When reacts with water, it dissociates into ammonium ion and hydroxyl ion. So here ammonia is acting as a weak base and ammonium ion is acting as a conjugate acid. So in both the reactions you see double arrow, right? This shows equilibrium, meaning the reaction goes both ways and it doesn't go to completion. Now let's see what is the degree of ionization and strength. The main difference between the weak and strong acids or bases is the degree of ionization. Let's see how. Strong acids or bases ionize completely. That is almost close to 100%. Whereas weak acids or bases ionize partially, that is less than 5%. So because of this, weak acids produce pure free H plus ions. So their solutions have a higher pH than strong acids and same concentration. If you see weak bases, they also produce pure free OH minus ions. So their solutions have a lower pH than strong bases or alkalis at the same concentration. Now let us study what are acid-base ionization constants. To measure how weak or strong an acid or base, we use equilibrium constants. The general formula for calculating the dissociation constant is denoted by Kd, that is equal to products by reactants. Now let's see how this formula applies. For a weak acid, Ha Now this Ha dissociates into H plus and A minus. So here the acid dissociation constant is denoted by letter Ka. So in place of D, we have written A. So here it is acid dissociation constant is equal to products. What are products here? Concentration of H plus into concentration of A minus by reactants. What are reactants here? Ha. So where Ka is equal to quantitative measure of the strength of the acid. Concentration of H plus ions. Concentration of conjugate base. Concentration of weak acid or undissociated acid. Let's see for weak base. For a weak base, B, when reacts with water, it dissociates into BH plus OH minus ions. So the base dissociation constant Kb. Again, products by reactants. What are products here? Concentration of BH plus into concentration of OH minus ions by reactants B, where Kb is equal to the quantitative measure of the strength of the base. Concentration of conjugate acid. Concentration of OH minus ions. And B is concentration of weak base or undissociated base. So by calculating this, if you get smaller Ka or Kb values, means those are weak acids or bases because they ionize partially or less. If you get larger Ka or Kb values, means they are strong acids or bases because they ionize completely or they ionize more. Now let's study what is the relation between Ka, Kb and Kw. Ka we have already studied, acid dissociation constant, Kb base dissociation constant. Now let's see what is Kw. It is ionization constant of water. So here the water molecules undergo self-ionization to form hydronium ion and hydroxyl ion. So here they undergo self-ionization to form hydronium ion and hydroxyl ion. H3 plus is hydronium ion. This you already know, hydroxyl ion. If you take Kw, concentration of hydronium ion and hydroxyl ion. The concentrations of both hydronium ion and hydroxyl ion in pure water is 1 into 10 to the power of minus 7. Therefore, Kw is equal to hydronium ion is 1 into 10 to the power of minus 7, and hydroxyl ion is also 1 into 10 to the power of minus 7. Then finally we will get Kw is equal to 1 into 10 to the power of minus 14 at 25 degrees Celsius. So here, what is the relation between Ka, Kb and Kw? Kw is equal to Ka into Kb. This for conjugate acids and bases. And this relationship helps us to calculate the strength of the conjugate acids and bases if the Ka is known and vice versa. Now let's learn how to calculate the pH of a weak acid. Since ionization is partial, we have to use concentration of H plus is equal to square root of Ka into C concentration. Then pH is equal to minus log concentration of H plus. Means we have to substitute this formula in place of concentration of H plus. Now the calculation for weak base. Concentration of OH minus ions is equal to square root of Kb into C concentration. Then pH is equal to minus log concentration of OH minus ions. Now these are the formulas we have to use when calculating the pH of weak acid and weak base. Now let's quickly revise what we have learned. First, weak acids and bases partially ionize in water. Second, they are described by their constants Ka and Kb, which means the strength. Third, you can calculate pH by using the formula for acids. Concentration of H plus is equal to square root of Ka into C, and for bases OH minus ions is equal to square root of Kb into C. And finally, we have seen the relation between Ka, Kw and Kb. Kw is equal to Ka into Kb, which is an important identity for conjugate acid base pairs. That's all for today's video. I hope you have understood the concept clearly, and if you have any doubts regarding the concept, please comment on your doubts. And if you find this video helpful, please like and subscribe to Biochemistry by Revati. And in the next class, we will cover the concept of biological buffers and their physiological importance. Until then, keep learning and stay curious. See you in the next class.